So our seminar speaker today is Yiannis Kaznessis from the University of Minnesota, and he's going to talk to us about multiscale models for new antibiotic technologies.
So very many thanks again to the organizers for the chance to present. And it's a privilege to work here with this group for the next few months. Allow me to just start with some general arguments. The main argument that I want to start making is that antibiotics is one of the most important discoveries in the history of humankind. Over the past 80 years, we have seen our life expectancies go up. The standards of living globally have improved markedly thanks to antibiotics. But bacteria are fighting back. They develop resistance to everything that we are using to fight them. There's a timeline from this paper in 2007 that shows on the top the time of the introduction of the various important classes of antibiotics. And what is disconcerting is that you can see on the bottom that even just a few years later, the first report for a resistant strain to any antibiotic class was reported in the literature. It only takes a few years before we observe bacteria that are resistant to any class of antibiotics that we have developed. So there's this pressing need to continue developing new classes, new molecules to keep on fighting bacteria. This need, I think, becomes even more clearly pressing when we take a look at these two trends. Yes?
I had a question on your previous slide. I don't know if we're going to get to it, but just the other comment besides the resistance is that the pace at which new antibiotics are being developed seems to be slowing down.
So
you're going to get to that?
Yeah, so this goes into the right-hand side graph, right? So we have these two trends, right? So the left-hand side is the percentage of bacterial infections in US hospitals with bacteria that are resistant, right, to our most prominent or most powerful antibiotics, including methicillin, vancomycin. So the percentage of these infections that are resistant inside US hospitals has been increasing steadily. At the same time, we have the decrease, right, the market decrease of new medicines, new drugs that have been approved by the US Federal Drug Administration, Food and Drug Administration. So on the left-hand side, we have evolution at work, right? I mean, if we pressure bacteria, they will inevitably always, right, respond back by developing resistance to the medicines that we use to fight them. On the left-hand side, on your right-hand side, we have this oxymoronic financial disincentive, right, at work. Antibiotics, right? You get a prescription of antibiotics, right? All 30 pills, right? The doctor will insist that you take all 30 of them, but after you take this one bottle, like this one prescription, in principle, that should be it, right? You should not need to go back. Contrast this, right, to medicines that must be or are prescribed for the entire life of a patient, like blood pressure or cholesterol-lowering drugs. I mean, pharmaceutical companies only have finite resources, so they will invest these resources, right, to develop drugs that will ultimately result in the biggest return on the investment. And antibiotics, right, simply do not result in high returns on the investment. So what we have been doing in my group at Minnesota over the past 12 years, and historically, just to give you a sense of where we're coming from, before going to Minnesota, I was at Park Davis, a pharmaceutical company in Ann Arbor in Michigan, and I was involved in their computer-aided drug design group and their structural bioinformatics group. That's where I picked most of the biology that I know. When I went to Minnesota, we started studying antimicrobial peptides. Right? So antimicrobial peptides are natural antibiotic proteins, right? So you can find the small proteins, you can find them anywhere you look in the biosphere. You're gonna find them, you know, on the trunk and the leaves of plants and flowers. You will see them, you know, you will find them in fish, you will find them in insects, you will find them in mammalian organisms, and we find them in humans, right? In our saliva, on our skin. They are really a first line of defense, right? Innate immunity line of defense, right? They kill bacteria. And what we wanted to do is to study these antimicrobial peptides, trying to understand the mechanism of action. How is it that they kill bacteria? We focused, right, over the past few years on a number of different classes of antimicrobial peptides, and protegrin is the one canonical AMP that we spend a lot of time trying to understand. It's just a small peptide. It's found naturally in the white blood cells, the white cells of pigs, pig leukocytes. And it is a remarkable peptide in terms of its activity. It just kills all bacteria practically in contact with protegrin will just cease to grow. We like it because it is small, right? And it will allow us to study it with the modeling formulas that I'll discuss in a minute. It's also very stable, right? So its structure is very stable in solutions, just simply because there are these two cysteine bonds. There are two bridges that just stabilize the structure of this peptide. What you see there, it's like a beta hairpin type of structure. Protegrin 1 or PG1 has also 6 arginines. These are positively charged amino acids, right, at physiological pH. So protegrin 1 carries a plus 6 charge at pH 7. Okay? So,
sorry, just one question.
Yeah.
Go ahead, Kostas.
So can I ask the very naive question, sort of like on the big picture? So when we use antibiotics, we say that bacteria are becoming resistant, so we need to come up with new antibiotics. So what is fundamentally different about these things that evolution has not sort of taken care of?
So in principle, so antibiotic molecules, right, are molecules that have specific molecular targets, right, in the bacterial cell. And bacteria, so there's the lock and key mechanism, if you wish. Here's the molecule, here's the protein, right, inside the bacterial species, right? It's just the molecule is designed so that it binds very strongly onto the protein, right? And it just stops the protein from doing whatever it is that it's doing, which is vital for the cell. So what bacteria can do, right? So these are proteins, and they can mutate them, right? And they can grow very fast, and they can mutate very fast, at a very fast rate, fast basis. So it's just in principle, right? I mean, a simple type of mechanistic understanding of resistance emergence is just a single amino acid, right, can be changed, right, in the structure in this protein that the bacterial species has, right, and your medicine does not bind anymore, right, so it becomes ineffective, right. I'll explain that these antimicrobial peptides, in principle, for a number of classes that we have studied, work in a different mechanism altogether.
So just, do I remember correctly that most antibiotics work only against gram-1 sign bacteria? And so this is, no.
No, so there are antibiotics that work against gram negatives, gram positives. There are antibiotics that are broad spectrum and kill everything, right? And gram positives and gram negatives vary, like on the molecular components on their surface, right? Practically. Okay, so without further ado, I'll tell you how we now know protegrin 1 kills bacteria, right? Which will bring me back to the lowering, of the likelihood that bacteria will develop resistance to this particular peptide. Right? So evolutionary also, if you look at antimicrobial peptides, right, they have been there for millions of years, right? And bacteria are still struggling to develop resistance. And largely this is why, right? Because protegrin 1 will bind, right? So it's positively charged, will bind onto the lipid membranes of bacterial cells, right? So the lipid bilayer of bacterial cells has a lot of, is rich in anionic phospholipids. So there's an electrostatic interaction certainly that will bring protegerine on the membrane of bacterial cells. Protegerine 1 will insert inside the core of the lipid bilayer. It will dimerize, oligomerize, right? And it will form pores. Okay? Let's take a step back, right, and look at what healthy cells do in principle. So healthy cells have high concentration of proteins, DNA, metabolites within them than their outside environment. So they're always under osmotic pressure. Water always wants to come inside the cells, right, and dilute the contents, right? And water can readily penetrate, go through this lipid bilayer, right? There's no transport barrier for water, right? It can go through. What cells will do, right, all cells will do, bacterial cells to be more specific, right? They will, using proteins, right, on their membrane, they will pump ions in and out, right? They will pump, so bacterial cells, E. coli cells, will pump sodium ions outside, potassium ions inside. Everything has to be electrostatically neutral, right? Otherwise things do not work. But there's this gradient of ionic species across the membrane that bacterial species, the bacterial cells actively maintain, okay? What this does is it creates what biologists call a transmembrane potential, okay? What this does, what you have to look at ultimately is the chemical potential of water. And there are two opposing forces on the chemical potential of water. There's the osmotic pressure, right? And then there's the transmembrane potential, okay? And the two balance in a healthy state, right? Balance one another. So the chemical potential of water is the same inside and outside the cell. There's no net transport of water, right? So the chemical potential is the driving force for transport. Okay, so enter these antimicrobial peptides. Bind, insert, oligomerize, form a pore. Right? There is a gradient, right? A concentration gradient across the membrane, right? So very quickly through these pores, potassium will leak out, sodium will come in. The transmembrane potential will collapse, water will come in because of osmotic pressure, blow the cells up. That's how it kills them. So what we have done is we've used a spectrum of different modeling formalisms to understand the thermodynamics and the kinetics, right, underlying all these different steps, right, for how antimicrobial peptides, how protegerine 1 kills bacteria. So I won't have time to go into the details, so I'll skim over the various modeling formalisms that we used.
Would this happen to all cells, not just bacterial cells?
So there is toxicity associated with these antimicrobial cells, with these antimicrobial peptides against mammalian cells at high concentrations. The mechanism is different, right? So mammalian cells will also maintain an ionic species, a transmembrane potential, right? These antimicrobial peptides, protegerine to be specific, cannot insert inside the lipid bilayer of mammalian cells. First of all, they're not rich in anionic phospholipids, and then there's a lot of cholesterol in mammalian cells that changes the rigidity of the membrane. So it's very hard for this particular peptides here to insert themselves. You put a lot of them, right? What they're gonna do is they're gonna form sheets, right, on top of the mammalian cell membrane, thin it out, and I mean that's how they can become toxic at very high concentrations.
Does this one work for anything with a cell wall, right?
Say that again?
Anything with a cell wall?
In principle, yes, right? So, I mean, like, there's a sweet spot, right, where you have enough AMPs, right, a high concentration to kill bacterial species, but not high enough to deal with, to kill mammalian cells. It'll come back, you know, to when we practically try to use those and deliver those. Do you have another? Okay.
Does it kill gram negatives or gram positives, or?
So the ones that are targeted with protegerine 1 are gram negatives. So we're talking about the inner lipid bilayer membrane. Protegerine 1 can go readily through the outer layer of gram negatives. So E. coli and Salmonella, which are gram negatives, are what we're using protegerine 1 against. So thermodynamics, kinetics. So for example, we look at the lipid bilayer, you know, from an atomistic point of view, all, you know, just atom type of models for lipid bilayers, and I'll explain this better in the next couple of slides. But we first calculate free energies, potentials of mean force, right? So the free energy of interaction between any two molecules, right, is really equal to the reversible work, right, that is necessary to pull them apart, right, to an infinite distance, right, within a solvent, right? So we can simulate this in a computer simulation where we have the membrane and we have the peptides, right? And we can pull the peptides, we can insert them, we can pull them, from inside. We can come up with different estimates for the free energy, which is the relevant thermodynamic quantity at constant temperature, constant pressure type of systems, like the ones that we study here. We come up with all kinds of insightful observations. For example, protein 1 will bind onto membranes, right? There are electrostatic interactions that stabilize this particular binding, right? But really, the free energy is lowered primarily, right, thanks to the release of counter ions. So there is this entropic effect, right? So membrane is negatively charged. There are ions around it, like sodium ions or potassium ions. When protein comes and binds, these ions, right, are released, right? And there's this entropic benefit, right, that minimizes the free energy more so than the enthalpic benefit of the actual Coulombic interactions. Right? So we have this type of insight that we can get with atomistic simulations. We look at, so that the biologically relevant structure is a pore. We know that it is a pore thanks to experiments, right? NMR experiments that Mayholm at Iowa State has conducted over the years. And we know that inside lipid bilayers, so protein forms very stable dimers, and then 4 or 5 of these dimers form an octameric or a decameric pore, right? So we have the structure from NMR, and then we can reconstruct the pore, right, within a lipid bilayer, right, with lipids that mimic the lipids found in E. coli or Salmonella cell membranes. So what we do is we run just brute force, right, molecular dynamics calculations here. So this is a system, 150,000 atoms. We can simulate for hundreds of nanoseconds. So I mean, we can start looking into events, right? Like water diffusing. We can look at the structural integrity, certainly of the membrane. We can look at the structural integrity of the pore, and we can look at the ions just moving through and through the pore, just calculating the different time scales that it takes for the transport of ions. So we measure, right, during the simulation, atomistic simulation, how many times we see chloride ions, how many times we see sodium ions or potassium ions, right, depending on what we simulate. The trouble that everybody has with brute force atomistic molecular dynamic simulations, right, is that the relevant time scales that you can reach. I mean, we simulate for months, right, just to go to what, 150, 200 nanoseconds, right, and we can observe like a few events, right, of ions going through the pore, but not very many. Okay, so we need a different modeling formalism, right? And we go then we use information from the molecular simulations to set up a continuum model for the transport of ions through pores, right? So these are Poisson-Planck type of equations where you have just the diffusion of the ions through the pore, right? Just because of a concentration gradient, and because of an electrostatic potential, right? The transmembrane potential that we can apply externally to our system. Of course, now when ions move, right, the electrostatic potential itself changes, right? This one can be calculated using the Poisson equation and the torque couple, right? So you have the Nernst-Planck, and then you have the Poisson equations, and we just integrate those in time and in space, right? Just to find at longer time scales what happens with ion transfer. Without going into the details, I mean, we can figure things out. The other thing that we can do here that we cannot do in molecular dynamics is apply just go into non-equilibrium calculations where we do have an externally applied transmembrane potential. You cannot do this in atomistic simulations. I mean, you would be just adding energy into the system that there's no way to take out.
Sorry, just one thing on those equations. I understand that the fact that they're both steady, you are instantaneously adjusting the ion concentrations potentials.
So this is a steady state. I mean, we've done this for kinetics as well. I'm just only showing the ones at steady state, and you can do this with different transmembrane potentials that you assume constant, or you can look at the kinetics, and this is what we do here. So we look at the whole entire cell now, right? So we look at the whole entire cell, and we model the transport of water, right? And so the transport of ions and the transport of water. Right? So we model the transport of the ionic species, right? Just based on what we get from the Poisson and Planck equations. And then we have the volume of the system, right, which is really the amount of water inside the cell, right? That we couple, I mean, the chemical potential of water inside and outside, and we couple this to the concentrations of the ionic species.
Is U electrostatic potential here?
So, which one?
U? E to Q I U. I'm just curious what U is.
No, this is not an electrostatic potential in there.
What is U?
This one should be mu, actually. So I think that it's a chemical potential. I don't remember right now what U is, what this U is.
It's coupling to a charge. Is Q a charge?
So Q is a charge.
If it comes from the
A is
Goldman,
the area, right, of the whole. So it cannot be a chemical potential.
if this is Goldman equation, this is the
So
voltage.
it is the Goldman equation.
Yeah, so this is voltage.
It's the voltage.
Yes. So you are not actually solving what you said about the PNP equations. You're not, I mean, analyzing them in this narrow pore, or are you?
Yeah,
Or you do?
they're very stiff, right? Very stiff. We use boundary conditions from the atomistic simulation. So we also use a charge density that is fixed for all of the rest of the atomistic, so the lipid bilayer, or the phospholipids, right? So we get all of this information from the MT calculation, right? So we have the charge density. We have an estimate for the dielectric constant as a function of the position x, y, and z coordinates, right? So that's what we do in that one. So you're right. The U here must be the potential.
And so what is the size of the pore, of the radius?
So the radius of the pore is about 40 angstrom, which is about the same as the thickness of the lipid bilayer.
So 40 angstrom. So are you inside the pore in a discrete regime where you have ion one at a time, or you are in the continuous one?
So we only allow, if I remember correctly, we only allow one ion at a time. I don't think that we do allow for.
So
I remember different things. So we've done a number of different things. So in the X and Y directions, right? So in the Z direction, right? In the X and Y direction, we have agreed with more than one point. So in principle, there can be more than one ions in there, if I remember correctly.
I thought your previous slide, you were expressing the ions in terms of concentrations, not discrete locations.
No, right. So they can be anywhere in there, but then when we numerically integrate, right, we have to discretize the space in there, right? And we have to just distribute the charges on the discrete. I mean, it's a finite different type of calculations that we run. So we have to discretize space. As I said, the cause of death. So we calculate transients of kinetic, the kinetics of the ion release. For chloride, for sodium, for potassium. And this is when we started doing experiments. This is when we went into the lab, right? And we can synthesize protegrine, right, with solid state synthesis. And we learn how to grow E. coli, right? And we were able to just use protegrine 1 against E. coli in cultures. And using potassium probes, right, we were able to measure the kinetics of potassium release from healthy E. coli cells upon treatment. Here I'm just showing the kinetics of the potassium release that we measured experimentally using an electrode, right, inside a culture. And then we find, with the examples from the culture, we find indeed, that the death, right, the percentage of cells that cannot be reconstituted, the kinetics of the death of bacterial cells is parallel to the kinetics of the potassium release. We also take SEM images, right? So this is E. coli, health E. coli before we treat with PG1, and then you see, just a couple of minutes later, right, after we treat them with PG1, they're bloated up, and their membrane integrity starts being compromised. They literally spill their guts. We have a fairly clear idea, right, of how antimicrobial peptides work. We have a fairly clear idea of how to engineer them, engineer new ones, so that they're active. We've modeled antimicrobial peptides against mammalian model membranes, right, just to figure out how things work there. Can we use antimicrobial peptides as antibiotics? Right? The short answer is no.
So what happens to the remaining 20%? No, it doesn't look like it'll ever reach 100%.
So for what?
Oh, sorry, I was looking.
So now
at
this
the
is
wrong.
so
I was looking at
I
the
don't have
wrong.
the units here, right? So
Yeah.
So the green line is for
Yeah. I thought the blue line was that I read it the wrong way round. I thought that was the
So the-
percentage killed.
So yes, it is the percentage scale. So the green line goes to 100%, right? So
I
all
read it
the
the other way. It's okay. It's okay.
So how many pores are we talking about?
So this is the
Roughly.
one. This is the one parameter, right, that we have no information from experiments on, right? And in order for us to match this kinetics here, we had to assume an average of about 100 pores per cell. Right. So these are all, of course, transient structures. Right? There's nothing equilibrium about these structures or about this set of phenomena. But on average, right, an order of magnitude of 100 was sufficient for us to fit the model results to the
And what sort of
experiment.
imbalance in the ionic fluxes between the sodium and potassium?
Say that again.
What sort of imbalance between the ionic fluxes in the sodium and potassium do you need for these bacteria to be killed in 2 minutes? How does it change?
So they change very rapidly.
No, but the ionic fluxes, how does it change? What sort of difference do you need in the ionic fluxes to kill the bacteria?
It happens, right, within a minute or two. So I'm not sure exactly what the question is. So this is what we get from the model.
Okay, I guess it's the y-axis. So,
Right.
it's the
So this is
concentration.
time, right, and this is the concentration. And we start with the concentration of potassium, sodium, and chloride, right, that we know is within a cell and then outside the cell in terms of the sodium, right? And I don't remember the numbers now. It's 0.15 molar sodium chloride is what comes to mind for the sodium chloride concentration outside,
Yeah.
usually, right? And I do not remember what the potassium chloride is inside the cell, but I think it is higher in potassium than in sodium, just simply because there are a lot of anionic species inside the cell, which of course, right, we do not take into account in terms of transport. Right? I mean, we assume that there's no transport of proteins or DNA or metabolites for that matter throughout the.
Yeah, no, I can see the ratios here, so you've answered the question.
No, the answer is no. Why? Because they're proteins, they're degraded quickly, right? I mean, you cannot orally administer peptides. People have tried this before they go to the stomach. I mean, the numbers just go down substantially. Right? They will be digested, right? You cannot inject anyone. People have tried this with animals with these antimicrobial peptides at high concentrations that you need for efficacy. They become toxic. They become toxic to red blood cells, to liver cells, to kidney. Nothing good comes out of just injecting anyone with this antimicrobial peptide. So here's the solution that we have been pursuing. Instead of delivering the antimicrobial peptides directly as proteins, what we have been working on over the past few years is cell buds. These are probiotic bacteria. Probiotic bacteria modified to produce and secrete antimicrobial peptides. If you consumed dairy products, say yogurt or milk, you likely consumed lactic acid bacteria. These are safe to consume bacteria. They're bile-resistant. They're very hardy organisms. They go through your stomach. They will go into your GI tract, right? And some of them will reside there. Others will just clear out in a matter of hours. We have isolated libraries, right, of these benign bacterial species, right? And we can engineer them, right, to produce proteins, right, but antimicrobial peptides, right? So the way that we do this, that is doable right now, is through synthetic biology. So it's fairly inexpensive, right, and the costs are going down to synthesize DNA. Right? So you take the protein sequence, you take it, as think of it as a DNA sequence, right? And it is 30 cents, right, to synthesize DNA, a base, right? So for a few hundred bases, right, base pairs, I mean, you need a couple $100, right, to have the DNA synthesized. You can insert that in a plasmid vector and take that plasmid vector inside the bacterial cells, the probiotic cells, that we use as delivery vehicles. Right? So we use delivery vehicles for antimicrobial peptides using this probiotics.
It's a good idea to take this as a supplement. It seems like it'd be a bad idea to kill off your internal flora of in there. It seems a bad idea to kill it off. Also, you can't undo it once it's started. You can't stop taking this and just have it turn off.
So, I mean, you can find probiotics in principle, on the shelf,
right? But they don't secrete stuff that kills off stuff as I do.
Right. So what you need to do then, right, in principle, is to have the probiotic produce a peptide that doesn't kill everything else. Right? It only targets the pathogen that you want to target inside the gastrointestinal tract, right? And of course, if you have someone who's really sick, and there isn't much else to do because the bugs are resistant to antibiotics, then you'll have to use this as like a final resort. How this impacts the microflora is certainly something that we're concerned with, right? The actual host set of bacterial species in their gut. I'll talk about this in a few minutes. Okay, so we've gone through different versions, right? So we've been trying to engineer these probiotics, right? And we've equipped, so Lactococcus lactis is one that you can find in Greek yogurt. It's terrific, right? That's why our mothers always say, just when you're sick, just eat yogurt, right? You literally reconstitute your microflora with this bacteria that are benign. They can help you digest, right? They can help you actually make metabolites, right, that are hard for your own body to make. Okay, we take them, we isolate them, we know how to manipulate them and engineer them to produce any one of these different peptides. We tried the expression. So the experiment that we have is we grow the pathogen, Salmonella, let's say, right? In a different beaker, we grow our probiotics. We can spin the probiotic, the cells down, right, and take just the supernatant, right? So the solution that contains the antimicrobial peptide, right, and use that in antimicrobial assays, right, to see whether indeed we can stop the growth of Salmonella E. coli. And so this is the growth, right? So this is what we measure here in time. We measure the growth, right, which is always exponential for healthy cells. And so with just a negative control treatment, and then when we treat with the supernatant, right, we just suppress the growth of Salmonella. So the peptides that are produced and secreted by the probiotics work against Salmonella in this experiment. I mean, we've done this with a number of different pathogens right now. I mean, a grave concern in hospital environments is vancomycin-resistant Enterococcus. So these are nasty pathogens. I mean, people get sick. With the help of collaborators, I mean, we've gotten our hands into vancomycin-resistant enterococcal strains that have been isolated from patients in the hospital. So we designed Lactococcus lactis to produce these different peptides that are specific, right, to enterococcal strains. Now, what is interesting is that these peptides are actually produced themselves by enterococcal strains. They use those, right, to control their own population density. They use them, when there is a dearth of nutrients, they use them, to just suppress their own population density. So they work very specifically against these enterococcal strains. We engineer promoters, right? So the promoter is, so you have the DNA construct with the antimicrobial peptides. In order for it to be expressed, you need to have a DNA promoter, right? The part of DNA that RNA polymerase will combine and initiate transcription, right? To make mRNA to then be bound by their ribosome to make the protein, right? The antimicrobial peptide. So we have built libraries of promoters, right? Different promoters that allow expression, that enable expression in different kinds of situations. So in this case, we have promoters that activate, that initiate expression of the peptide when the chloride concentration in the medium is as high as it can be found in the stomach and the upper gastrointestinal tract. Another set of promoters that we've built are promoters that respond to the pathogen itself. Right, so you have the probiotic going inside the gastrointestinal tract, and it produces nothing unless there is a signal that comes from the pathogen itself. Right? So what we did is we went to enterococci, and we found systems that they themselves use to communicate with one another, quorum sensing type of systems. Right? And we hijacked a system called PERTS-X, PERTS-Q, and we took it from enterococcal strains and we inserted it, we engineered Lactococcus lactis, right, to respond, right, to this pheromone molecules that enterococcal strains produce. And it is only when this enterococcal pheromones, the concentration in the medium is high enough that then there is the production of the enterocystin that goes back and kills the enterococci. Nasty pathogens, as I said, we've tried, so these are growth curves of these enterococcal strains. So the black one is just, in media with sugars, right, so that they grow happy. But then we start adding, ampicillin, vancomycin, streptomycin. They don't care, right? Just they keep on growing. Maybe, a little bit of slow, just a delay in their growth, but ultimately, they're really resistant to these antibiotics. When we use here, just the supernatant again from our culture with the modified probiotics, we see suppression. So I want to come back to modeling and how modeling has helped us engineer, just guide us, in this process of engineering synthetic biological constructs. So one of the motivations is that this PERTS-X, PERTS-Q, right, and I won't go into the molecular biology of it, but it's ultimately a response system, right, that shows exhibits a bistable behavior. There are non-linear interactions in here, right? So we had to model it to figure out how exactly this works and how we could best engineer this in lactococci. How do we do this, right? So in principle, when it comes to models of these synthetic biological networks, what we do is we adopt Jacobs and Monod's postulate that however complex, the behavior of biological systems can always be reduced into cascades of molecular interactions. So we take all the interactions that come from the molecular biology dogma, transcription, translation, right, protein degradation, protein folding, protein-protein interactions, and we model all of those. And ultimately what we do is we have networks of reactions, right? Network of reactions that we model the kinetics of. There's no need for me to expand on the virtues of stochastic modeling, right? For these reaction networks. They are away from the thermodynamic limit in biological systems, right? So and this is why we've developed a number of tools, right? So hybrid simulations for stochastic discrete, stochastic continuous type of modeling formalisms combined. We make our software available. I should just go quickly through the first example because I think it's fairly straightforward for me to describe in a short amount of time, where we use these models to guide us into engineering synthetic biological constructs. We build an end gate, right? So this is just a parenthesis, right? It doesn't have much to do with doesn't have anything to do immediately with the synthetic probiotics, right? Producing the antimicrobial peptides, but it gives you an idea of how we model the systems. So we wanted to engineer E. coli to function like end gates, logical end gates. E. coli should give out a signal, a green fluorescent protein that we can see, we can measure using fluorescence experiments, right? But they should only do this if and only if there are two molecular species that we add inside the culture. So we're growing them, right? And we have to add this IPTG, which is a small sugar, and ATC, which is a small organic molecule. Only when we add these two molecules, right, should the E. coli produce this GFP. How do we implement this? This is practically it. So this is the DNA sequence, right, that we engineer. This is the green fluorescence protein that, when transcribed and translated, will give us the signal that we want to get us output. So here, the promoter is what we really engineer in this particular case, right? So bacterial promoters, right, have this -10 and -35 positions, and these are really 4 or 5 nucleotide base stretches of DNA, on top of which, right, RNA polymerase comes and binds, right? So it recognizes, binds very strongly, and then initiates transcription. Right? It starts recruiting tRNA, just stitches together mRNA, right? Which then goes into translation. Okay? What we can do is certainly take the -10 and -35 positions and have around them these operator sites. These are again DNA sequences, specific sequences, right? The lactose operator and the tetracycline operator, the L and the T in the system. So these operators are recognized by transcription factors. These are proteins, right, that are inside the cell. So these proteins, right, will find and bind this operator site. So the tetracycline repressor will bind on the tetracycline operator. The lactose repressor will bind on the lactose operator. Okay. So here's how the end gate functionality works. Right? If there is no ATC or IPTG, then these molecules will be on the promoter. RNA polymerase cannot bind, right? Just simply, just physically excluded, right, from the promoter. So there's no transcription. When we add ATC, what ATC will do, right, is it will bind the tetracycline repressor, and it will induce a conformational change. A change in shape that disallows the tetracycline repressor from binding on the DNA anymore. So it just releases DNA. The same goes with IPTG. IPTG binds on the lactose repressor, and the lactose repressor then releases DNA. And so you can see probably how the end gate works. Right? If I have none or just one of these two molecules added, there will be two or at least one of those repressors on the promoter. No output. Right? Only if I have both of those, right, ATC and IPTG, will all the proteins be released and the promoter be free for expression. So this goes back to what I was saying about cost, right? So we can engineer, this is the sequence, right, that we, across the street, I sent an email across the street from my department in a DNA synthesis facility, right? And 2 days later, they send me a vial with this DNA, just $100 per system, right? So this is the actual sequence for the LTT system, right? So we take that and we insert it into this plasmid. These are circular DNA constructs, right? That we can then insert into the bacterial species that we want. So DH5 alpha pro are E. coli that produce the lactose and the tetracycline repressors constitutively. So these proteins are always inside the system, right? So we can do this with what? We can do this with LTT, we can do this with TLT, TTL, and then have double L operators, right, in single. So we have multiple designs that we can try, right? Because we can mix and match. We can take this part here, right? We can take the sequence for the lactose operator and just move it downstream, like past the -35 or past the -10 position.
What restricts you from just having, for just having 3 segments?
Why do I not have more, you mean?
Why 3?
So there are these 3 positions. So geometrically speaking, RNA polymerase will bind, right? So you can have additional stuff further upstream. They're not going to be as effective, right? And actually, what we find is that this position and this position really determine much of the behavior that we observe.
Can I ask a naive question? When you get these in a vial, what do you actually have? I mean, not one strand of DNA. Like you have a solution with, like when you spend $100 or so, like what do you get?
Pure DNA, double-stranded DNA with that sequence. And the purity, it can go to 95, 99%, right? Easily. They synthesize this, right, from scratch. It's one base pair after another.
You have a ton of it.
I can have a ton of it, right? Yes. And then what I can do is just do PCR, right, and make even more tons of it, right? So.
So can I continue with the naive? Thank you, Dave. So I can continue with the naive questions. Okay, so this is a substance which is sort of like you have in a solution.
Yeah.
So what do you put that thing into the bacteria?
No.
I mean,
So this one we
I like
insert.
that you laugh, right?
Oh,
So cloning 101 without getting too technical, right? We have ends here, right? So this is double-stranded, right? With ends just hanging, right? That will go and bind onto the ends over here, right? So you have a matching like end on each side of this DNA that will go and bind onto this. So there's a process, right, that involves opening this up, right, with a ligase. So you use enzymes, right, to open DNA and then bring them together.
The DNA within the bacteria.
Right. So you have this, right, and you have this, right, and then you have enzymes, right, that will open up the circular DNA, right, and then stitch these positions here, right, and then what you do is you really take the cells, right, and you mix them with now your complete circular DNA that includes this sequence, and you shake them fast, at high temperatures, right? So what happens is that the circular DNA is uptaken through the membrane, right? So transformation is what this is called, and then you can select, right? So you should only have the bacteria that have this circular DNA in them, right? Because there will be others that will not, right? But the way that you do this is you use an antibiotic. So ampicillin resistance, right, is something that you encode in this. So when you use ampicillin right now on the culture, only the bacterial cells that carry this guy, the full one, right, will survive. And then you pick them up easily because the rest of them are dead.
Eventually they kick out the plasmid though, right?
They can, right? So I mean, the stability of this plasmid is something that we've studied, and it takes weeks for them to just keep them out. So even without, this is the important thing, even without ampicillin, right? So this is a fairly stable vehicle. So through many generations, over days, I mean, the plasmid stays in there fairly stable.
And hitting it with the antibiotic doesn't affect it long term
No.
at all?
Though the antibiotic will.
I mean, it kills the other guys, but like.
Not these ones. These ones have the resistance in them.
No, okay, but there's resistant, you don't die, but it doesn't change anything.
So it may inhibit their growth, but again, I mean, we will be able to pick them out, and then just put them in fresh media without the ampicillin. They have it inside of it. Modeling all the components, right? And we can do this, and I'll say this right now. We can only do this. We tried for a number of different systems, but we can do this with the detail that I'll discuss right now for the tetracycline and the lactose system, because arguably they are the best studied biochemical systems ever. So in terms of kinetics, in terms of interactions, there isn't a lot of room for us to improvise. So we have all this dimerization, the binding on the operators, the transcription initiation, translation. So we assume there is a pool, of components in the cell with a lot of RNA polymerase, with abundance of these molecules that we need. 63 reactions, not a simple system, right? But not one that we cannot not model. So we model this, and this is what we do. We have different initial concentrations of ATC and IPTG, right? And we measure the green fluorescence protein at the end. So we have a grid of input concentrations. We can model those for different designs, as I said, right? Just mixing and matching the different operator sites.
These are not spatial.
No. So these are homogeneous, right? There is a difference in those, right? In the way that they bind on this one versus this one versus this one, right? And this is the one type of parameter that we need at the end to fit. So kinetic simulations, probability distributions, right? We get the mean. Right? So this is a simulation result for one initial concentration of ATC and IPTG. Right? We take the mean at steady state and we put it here. So we have a grid, ATC and IPTG, the mean fluorescence, right? The output, the number of molecules that we have in the system. And then we come up with these two-dimensional surfaces and this three-dimensional plots.
And the kinetic rate, I mean, you have good estimates of these? Or
Yeah, so I said that.
Or you guess them or what?
So there's a few guesses in there, certainly, right? I mean, in terms of orders of magnitude, like the transcription in E. coli, translation in E. coli, and the binding on the operator side. So there are no other systems that are as well studied as the tetracycline and the lactose. And this goes back, right? And I'll comment. I hope I have time to comment on this. So biological system modeling, right, is nowhere near as mature as physical and chemical systems modeling, right? And a major difficulty is the absence of kinetic constants, of equilibrium constants. An absence, just a handle, right? How do I even start assuming values, right? Order of magnitude values for these kinetic constants. We didn't have to do this with this one.
So and even the ones that you sort of have to guess somehow because it works, that's kind of a verification that you're doing the right thing.
You can say so, although if we have to start guessing a lot of parameters, what is it, 12 parameters can fit an elephant, right? So
Yeah, exactly, because you had 63 reactions, so these are 63 parameters. Yeah?
No, I do not have 63 parameters, right? So I have to have a number of parameters that are order of magnitude assumptions that I make. But again, in bacterial species like E. coli for the systems, there is literature that will guide us, right, to get the order of magnitude correct. Experiments. We do the same thing in experiments. Flow cytometry, right? This wonderful technology. You take a culture of cells and you isolate them one by one, and then you measure the strength of fluorescence that comes out, which we assume is proportional to the number of pre-fluorescence protein molecules in there. Here's the result, right? So I'm showing only 4 different designs here: models, experiments. We can make a number of different comments here. So this one is not a high fidelity end gate, right? So at 0 IPTG, right, there is already output. And this is just because the lactose operator just unbinds even in the absence of IPTG. It's a leaky system, right? So, and again, we knew this from the biochemistry. This is the highest fidelity, right? And gate in the sense that there's little output whenever there is only one of the inputs. But you can see the end gate-like behavior. Okay, so what the modeling does is it helps us feel comfortable about what we see in the experiment, right? And it helps us just make decisions, right, on moving forward, right? It does not allow us, and I'll say this clearly here, we cannot use modeling, right, to design biological systems with certainty, right? We can use modeling, right, to design a mission to the moon or a mission to Mars, right? And be comfortable about what it is that we're doing, assuming that we're not going to mix up the different units in there. But for biological systems, we cannot do this. Okay? Only a posteriori that modeling, it just makes us feel good, gives us explanations about what we see experimentally. Let me finish, right? And I only have a few minutes, right, to do this. So we engineered, going back to the probiotics, using what we know now with TET-on, all these operators, we engineered the promoter in E. coli Nissel 1917. So this is an E. coli that is a probiotic strain. It was isolated from a German soldier and the First World War, he was the only one in his platoon that did not get sick from diarrhea, salmonellosis. So from his feces, right, this E. coli strain was isolated and was found, right, to work well against salmonella. Okay, so we took that, right, and we boosted it up. We engineered it to produce proteins, right, this microcin, antimicrobial peptides, that kill salmonella, right? So we have this vehicle right now to kill salmonella, producing even more antimicrobial peptides than it would do naturally. So we tried against salmonella, we see that it works, right? What's the next step beyond in vitro results in the lab? The next step is to try this in animal experiments, right? So here's the animal experiment that we ran. Poultry, right, is the #1 source of protein globally. Salmonella is the #1 pathogen, right, in terms of foodborne illnesses. Poultry are the #1 source, right, of salmonella pathogenic strains. What we wanted to do is to silence birds. Turkeys are of importance in Minnesota because Minnesota is the #1 processing and producing state in the states of turkey meat. You go outside Minneapolis, there's the one turkey farm after the other. It's just spectacular to see. So we get turkey birds day of hats, right? So we bring them. So Brittany did most all of the work that I'm going to describe in the next 2 minutes. 200 birds. We put them in isolation units, right, in St. Paul with the help of vet scientists, like, and animal scientists, right? Like, I have collaborators who help me teach us, how to have the birds fed and growing healthy, right? So we go 2 days later, right, and we challenge all the birds with salmonella, right? Just open their beaks, and just drop a meal of culture full of salmonella and Candidioides that, by the way, was isolated from an illness and humans getting sick from consuming poultry products. Right? So we have this one strain. The birds are not going to get sick. Right? The birds will be healthy, just happy carrying the salmonella around. Then we go the following day and we give them the Nisl, right, that is boosted up with the microcin. We go in different days, right, and we take birds from the different groups. So there's a group with salmonella and nothing, right? Salmonella and unmodified Nisl, salmonella and modified Nisl, salmonella and then an antibiotic, right? So we have these different groups. So we go get different days and get different 5 birds from each one of the groups. We've learned how to euthanize them and necropsy them. And inside the GI tract of birds, poultry, are the cica. So cica are these pouches, right, full of liquid, where there's fermentation going on. So birds will, all birds, right, will ultimately digest and ferment their foods inside their cica. And it is in there, right, that salmonella resides in the highest counts. It is this part, right, that if it bursts during processing, just spreads the salmonella all over the meat. Right? Salmonella is in there, so this is where we wanted to measure the salmonella counts. And ultimately, this is what we get. So here, salmonella counts per gram of cecal samples. Right? And these are days post the salmonella challenge. So what we see here, this is log, right? So these are orders of magnitude. So what we do is we measure the amount of salmonella, the counts of salmonella inside the untreated birds, and then birds, the green line is birds that are treated with the unmodified Nisl. In all of the birds, salmonella will clear out. Right? I mean, in 2 weeks, there's not going to be much of the salmonella that we inserted just once on day 2 of the experiment. But what we see is that when we use the modified probiotic, the salmonella counts drop, right, by 2 orders of magnitude, and they drop much more quickly inside the birds that are treated. Where are we going with this? again, we have libraries of probiotics, libraries of DNA promoters, libraries of antimicrobial peptides, and we choose different pathogens right now. So enterococcal strains, Clostridia strains are of interest in at the National Institutes of Health. And we want to engineer safety components, so components that will eliminate the chance of any one of these bugs being released into the environment. Last slide. I said this, modeling is not the same in biological sciences as it is in the physical and the chemical sciences, but it does, this is my argument, it can be a very creative source of knowledge, right, of insight that you get, right? Just by asking questions, right, what is the model that I should use, right, and trying to answer them, you get insight that is useful ultimately to take the leap, right, and start doing experiments, right? There's no rational guiding by models of the experiment, right? But what it does is ultimately just gives us the insight that we need to just move forward without just being blindsided very quickly. A number of students have contributed. Brittany did a lot of the work of the experiments. Kat worked a lot on the end gates. Dan did a lot of the work of the model. So I'll stop here by thanking you for your attention.
Sorry, I want to come back to the AND gate. So I guess the first question, can you remind me why you wanted an AND gate in this? And then secondly, so is this a first AND gate that was created from this kind of regulatory network, or is it the first AND gate that sort of worked in this context? I'm just trying to understand where this fits in the broader scheme.
So the end gate has nothing to do with the antibiotics.
Okay.
Right? So
Fine.
this was something that we did, right? For us to see that indeed when we model systems, right, and we engineer systems, right, the two have some relevance to one another.
Okay.
Right? Ultimately, this is why this was done. This one is it had never been done with these two components, right? People had built synthetic logical AND gates and OR gates, right, and NOR gates before, not with these components. Yeah.
Have you thought about targets not necessarily to kill the bacteria, but to hinder it in some way from infecting poultry or?
Yes, there are a lot of virulence factors, right? So bacteria will use, for example, proteins on their surface to recognize host cells and just dock on them, right? And they form biofilms, right, in the different parts of the gastrointestinal tract by recognizing receptors on host cells, right, and just binding on them with their own protein receptors on their membranes. Not us, but people have thought about making molecules that will hinder this particular type of interaction. There are other virulence factors. There are other proteins, right, that bacteria have. That if you stop, you can stop the path for disease, right? Just for them to translocate, for them to start producing toxins, for them. There are different ways that disease is manifest, right? You need to have a hold with high carriage, high concentration, and from then on, depending on the pathogenic strain, there are different ways that they can cause disease, and you can stop along all of these different steps that ultimately the cause of disease. We haven't done anything like this in this particular case.
Yeah, it's a very nice talk. At the end, you mentioned suicide genes, which I don't know anything about. Can you say something very quickly about?
Right. So you can have genes inside the probiotic, right, that will, upon atmospheric oxygen concentrations, right, result in the death of the probiotic itself. So the minute that it is released, right, and it uptakes oxygen from the atmosphere, because in the gut, the environment is to a large extent anaerobic, right? So the concentrations of oxygen are much lower than in the atmosphere. So you can manipulate, you can engineer a bacterial strain, right, to kill itself by producing, it can produce antimicrobial peptides that kill itself, right? It can stop producing immunity genes to the antimicrobial peptides, but it will do this, right, only when there is a lot of oxygen, for example, right? Or when the pH just stops being low. So there are different switches that we can use to eliminate the probiotic from ever really making it outside.
What about when they lose the plasmid? Like if you just want the bacteria to vanish after they've served their purpose.
So you can engineer the plasmid, right, to be dispelled at a certain point in time, and then it becomes the missile found in the gut of the German soldier, which is benign.
Yeah, right, that's true.
Okay. There are no other questions. Let's thank Yiannis again. So, when announced